at a greater length. So what is the genetic code exactly? The genetic code refers to the rules or the established rules that govern how the nucleotide sequence of DNA is actually translated into a sequence of amino acids in a protein or in a polypeptide chain. Now, during translation, the sequence of nucleotides in the mRNA molecule is actually read consecutively in groups of 3 nucleotides at a time. And that grouping of 3 nucleotides is what we refer to as a codon. So a codon is basically a triplet of nucleotides or a nucleotide triplet, and each codon will only specify one amino acid. So one codon will code for one amino acid. Now, it's important to remember that during translation, the mRNA strand is actually read from a 5' to a 3' direction, or in a 5 to a 3' direction, and hence, just like transcription, translation is also a unidirectional process. Now, because RNA is a linear polymer and because it's made up of 4 different types of nucleotides, there are therefore 4 to the power 3 or 64 possible combinations of the 4 different nucleotides. So if we need 20 different unique codes for the 20 different amino acids we have, it's clear to see that we should have more than enough combinations of these nucleotides or these codons to code for the 20 amino acids. Now, because we only have 20 amino acids, but we have 64 possible codons, it means that either some of these codons, some of these nucleotide triplets are either never used, or the code is redundant, and some amino acids are actually specified by more than one triplet. It just so turns out that what is actually true is that some amino acids are actually specified by more than one triplet. Okay? Now, the table that we have on the screen right here shows the 61 of the 64 possible triplet combinations that we have for amino acids. Now, the 3 codons that we can add to the 61 to make 64 that actually don't code for any amino acids are called stop signals or termination codons, and they're used to mark or signal or trigger the end of translation. So they're used to stop the translation process. Those codons are UAG, UAA, and UGA. These are what we collectively refer to as the stop codons. Now, when several different codons specify one amino acid, the difference between them usually lies at what is the 3rd base position, at the 3' end of the mRNA strand. So, for example, alanine is coded by the triplets GCU, guanine cytosine uracil, GCC, guanine cytosine cytosine, GCA, guanine cytosine adenine, and GCG, guanine cytosine guanine. This redundancy, in which we have one amino acid being coded for by more than one codon, is actually characteristic of the genetic code. Now, the first 2 letters of each codon are the primary determinants of what amino acid is coded for, and the difference between 2 codons that code for the same amino acid is usually at the 3rd position of the codons. I'm going to repeat what I just said, okay? Now, so the first 2 nucleotides, okay, in each codon, those are what will mainly determine what amino acid is coded for. Now, the difference between 2 codons that are coding for the same amino acid is usually at the 3rd position, at the 3rd nucleotide position of those codons. Now, I want you to take note of the fact that the codon AUG has a dual function. You will see it coding for the amino acid methionine, abbreviated MET, and which also functions as a start signal. So anytime you see the amino acid or you see the codon AUG, you know that that typically is a signal to start the process of translation. So genetic messages typically will begin with mRNA or the codon AUG, which then will signal the protein synthesizing machinery to begin translating the mRNA at that location. at that location at which the codon AUG occurs. Now, because AUG codes for methionine, polypeptides usually begin with the amino acid methionine when they are synthesized. Now, we have noted that we do have